All right, so when we look at the cell cycle, it's really important for us to realize that mitosis is only a really, really small part of the cell cycle. The most or the majority of our cell cycle is a process known as interphase. And interphase has quite a few different groups. So if I try and highlight it so that it lines up here, we have our G1 phase, which in my diagram here is orange. We have our S, or our synthesis phase, which is green. And then we have this really awkward brown color, which I'll use pink for, known as G2. The process of mitosis is just this really small blue part in this diagram here. So this is mitosis. We also have one other phase known as G0, and we will talk about that in a minute, but that's just where our cell hangs out normally. Because most of the time, again, our cells are not dividing. And this is what we can refer to as G0. That looks like 6. So continuing to look at each of these stages in more detail, I really like this diagram. Apart from the mitosis where it has multiple stages, we're only going to learn 4 stages for mitosis. So I just want to highlight that before we start talking about interphase. So each of these phases here, G1, S, G2, and G0, are all really important for us to learn. And then when we get to M phase later on, this is when we talk about our mitosis. So let's start up the top and talk about G1. What does the G stand for in each of these stages? It stands for growth. It is growth phase 1, G1. During growth, the cell produces new proteins, the cell grows, and the cell just exists. It does all of the normal tasks that it is designed to do. So if we spoke about a red blood cell, what's the purpose of a red blood cell? It is to transport oxygen, transport carbon dioxide, thanks to hemoglobin. This process ends when our DNA begins to replicate, when we want to create a new cell. Our next phase is the S phase. And my S here stands for synthesis. Synthesis is a fancy word for creation, and the reason that we call it the S phase is because our DNA is being replicated. We spoke about that at the end of our green booklet, where we take one long piece of DNA and we can turn it into 2 identical copies. Once we've copied our DNA, we then move into the G2 phase, into our second growth phase. And this second growth phase is our simple prep, or our preparation for our division that occurs in the M phase or our mitosis phase. Our last phase can be a little bit confusing to understand. Hopefully it will be a little bit clearer by the end of today's lesson. But when we talk about the G0 phase, it's not GO, but G0. This is when the cell leaves the cell cycle. It's no longer going round and round and round and making new cells, and it can stop dividing for extended periods of time. When we stop dividing, if I scroll up quickly here, we can see that these cells, once they're specialized, can hang out, not divide, and do a job, say like if we look at our kidney here, for a very long period of time. This is G0. It is not dividing. Then, eventually, we're going to restart up and we'll go to G1, where we might produce some new proteins and things like that. That's also going to be a part of our cell cycle. So, the only phase that we haven't spoken about in this diagram is this thin one in here that I'm highlighting in green now, called cytokinesis. We will talk about cytokinesis today, but I highlight it here to show that it is not a stage of mitosis. There are 4 stages to mitosis, and we'll talk about them in a minute. So, continuing on with our discussion of the cell cycle, we're going to start with interphase. I'm going to make it clear up the top here that interphase is not a stage of mitosis. I'm going to highlight it again. So interphase is not a stage of mitosis. It is simply, again, that period of time between cellular divisions where we go from our growth phase 1, apologies, that's a typo on my slide there. So growth phase 1, our synthesis phase, and growth phase 2. Now when we have a look up at our cell here, we can see clearly that we have all of the really important things for us as we dive into the process of mitosis. These include centrioles, a really important organelle for us. They include chromatin, which is our condensed or uncondensed version of DNA. And we can also see things like our nuclear envelope, which is going to be really important for us to identify different stages of mitosis, whether or not there is or is not a nuclear envelope. Scrolling down, this is our cell in interphase. So if you're following along in your booklet, I'd probably jump to page 9 now. This image up the top here that I'm circling, this is the one from your booklet, and you might like to write next to yours that this is a cell in interphase. During interphase, we have the following. We have DNA replication. Now, DNA replicates, as a reminder, during which of our 3 main phases of interphase? We've spoken about G1, S, and G2. Which phase am I looking for here? Great. I'm looking for S. I'm looking for that synthesis phase. This is where our DNA replicates. It is important to note, however, that DNA is not in the form of chromosomes during this time. We see DNA instead as chromatin. As we move down, we are now in the first stage of mitosis. So I'll write that up here, or I'll just write a 1 and I'll circle that. We are now in the first stage of mitosis. We are going to travel from P to M to A to T today, and right now we are in our P stage. So PMAT is a nice acronym that has commonly been used to help remember those stages of mitosis, and that'll really help you out when we get to meiosis as well. So our first step here, I'm going to switch over to blue to make it a little bit easier to see. Our nuclear membrane breaks down. So this border going around here, our nuclear membrane begins to break down, begins to dissolve. It might be less clear. I would say in this diagram it could be a little bit clearer. So you might like to include some dots or something like that just to show that it is beginning to break down, or label it in a way that says that the nuclear membrane is breaking down or dissolving. So you might draw a line out like that. During this stage, it's important for us to note that this DNA, or our chromatin, condenses into chromosomes. So it's during prophase that we have the formation of chromosomes, and they become visible to us if we're looking through a microscope. The last thing for us to note of importance during prophase is that our organelles, called centrioles, move towards opposite poles of the cell, and they start to produce what we call the spindle or spindle fibers. They're not shown in this diagram, but what you might do is you could show two, oh, that's a very bad color for it. If we just draw in, and we could draw some circles here and just label them as our centrioles. And they're moving towards opposite ends of the cell. Now that might be north and south for us. It could also be east and west, or it could even be somewhere along the line. As long as they're on opposite ends of the cell, that is the most important thing for us. Moving down, we are now into the second phase of mitosis. This phase, metaphase, you can simplify and remember simply as The M from metaphase is where we move towards the middle of the cell. And we can see this most clearly with our chromosomes lining up really nicely for us in this diagram along the equator of the cell. Now, I know this is some geography, I know it's not ideal, but equator of the cell is how we typically describe where these chromosomes line up along. During this time, when they line up down the equator of our cell, we can also see that these spindle fibers have attached to the centromere. So I'm going to label some things on my diagram here because centromere and centrioles are often misrepresented, often mislabeled. So using purple doesn't work. Using yellow, I'm just showing where my chromosomes are connected. If I draw this line out here, this is what we refer to as the centromere.
Do you mind moving
Our second thing of note, and I'll use my blue pen to circle around these, at each pole of the cell, we've just spoken about them, they are our centrioles. So a reminder, our centrioles produce our spindle, and our centromere is the part of the chromosome where they attach. So to recap, during metaphase, our chromosomes move towards the middle of the cell. Our centrioles have produced our spindle, which is attached to each chromosome pair at our centromere. We now move into the third phase of mitosis. And this phase here, anaphase, similar to mitosis' second phase, metaphase. Anaphase, we can take the A from anaphase, and the A from anaphase we can use to write the word away. Simply put, during this stage of mitosis, that spindle or those spindle fibers contract. Key word there, I'll underline it. They contract and they pull the chromosome apart. They pull it apart at that centromere. And we can see that they've now moved from our midline and they have begun to move towards the pole of our cell. Those sister chromatids we now refer to as chromosomes. So in our last diagram, we referred to the chromosome as both of our sister chromatids together. Now, when the sister chromatids are pulled apart, we still refer to them as chromosomes. During this stage, the last little bit, we can see that they are pulled towards the opposite poles. And what I'll do again is I will label that one in blue. These here are our centrioles. Our final stage of mitosis, we're going from prophase, metaphase, anaphase, we get to our last stage today, which is telophase. During telophase, all that I'm going to get you to know is that our nuclear membrane reforms, and it reforms around the chromosomes. Telophase is often lumped in with another process that we know as cytokinesis. Now you'll remember at the beginning, I highlighted cytokinesis on my diagram in green, and I said that cytokinesis is not a stage of mitosis. That is correct, and it is often lumped, unfortunately, with telophase. During cytokinesis, we have the division of the cytoplasm. So we can see this pinching in the middle of our diagram now. Eventually that pinching will form off and we will end up with two identical daughter cells coming from our one original cell. So the final result, after we've undergone telophase, where our nuclear membrane has reformed, and we've undergone the process of cytokinesis, we end up with two identical diploid daughter cells. So let's break that down. 2, the number of cells that are produced. Identical, they share the same genetic information. Diploid refers to how many chromosomes we share. So diploid is going to be our 2n number, and in humans 2n is equivalent to 46 chromosomes. Not all organisms have 46 chromosomes. The number of chromosomes differs based on the organism itself. So to recap, this here is taken from your book, and your book really clearly outlines what happens during each stage of the cell. Now, what we've gone through is pretty much exactly the same as what happens here. The only thing that I would get you to note, and perhaps add to your notes, is going to be surrounding telophase. During telophase, it is worth noting that our spindle disappears. We have the division of our centrioles, and our chromosomes uncoil and disappear. Now again, I'll highlight cytokinesis in green because it is not strictly a stage of mitosis, and I'll use my pink here to show interphase as well because it is not a stage of mitosis. These 4 in the middle here, prophase, metaphase, anaphase, and telophase, are all my stages of mitosis. Question?
When it says like centrioles divide, does that mean that each cell just has one centriole, or does it duplicate?
So generally we're going to have one and then it can divide in order to become multiple. Yeah. So looking down now, here is a real life example of a cell. Now this isn't actually a real life example, it's just a really good diagram. And the reason that I like this diagram is because it shows really clearly what a centriole kind of looks like, and it shows really clearly those spindle fibers coming out. So if we start over on the top left, we have our prophase. In prophase again, our chromatin coils and we get the formation of our chromosomes. Our nuclear membrane breaks down. Our spindle fibers grow out from our centrioles, and our last part here is that our centrioles begin their migration to opposite poles of the cell. During metaphase, step 2, chromosomes lie along the midline of the cell. I'm going to underline that word midline. So metaphase, we head to the middle. And what happens during this stage is that our spindle fibers begin attaching to the centromere, the thing which is holding our chromosomes together. During our 3rd stage, anaphase, our centromeres divide into 2. Our spindle fibers pull our new chromosomes to opposite ends of that cell. And now, each pole is going to get an identical set of genes. We are getting an identical copy of what we started with. Our 4th and final stage of mitosis, chromosomes gather at each pole of the cell and they uncoil, they unwind. We end up with our chromatin once more. Our new nuclear membrane reappears. We get a new nucleolus appear in each nucleus, and our spindle fibers disappear. Now, this image here also shows that we're beginning to get this pinching in the middle here, and to keep it consistent, I'll use my green actually. Keeping it consistent, this green is showing that pinching of those 2 new cells, and that is our cytokinesis, being our division of cytoplasm.